Hello, I'm Shriya. Today I'm going to talk about an American biochemist, Dr. Roger Kornberg. Dr. Roger Kornberg was born in 1947 in St. Louis, Missouri. He was the first of the three children born to Arthur Kornberg and his wife Slavy. With both parents being well respected scientists, it was not surprisingly that Roger Kornberg also developed an interest and enthusiasm for science. As I said in an interview, science was a part of dinner conversations and an activity in the afternoons and on weekends. Both my parents had fine scientific minds and taught by example how to approach questions and problems in a logical, dispassionate way. Scientific reasoning became second nature. Above all, the joy of science became evident to my life. Kornberg studied chemistry and biochemistry and without having to think about it became a scientist. He graduated from Harvard University in 1967 with a bachelor's in science and went to Stanford University for graduate work. His doctoral thesis was on the chemical nature of phospholipids. In 1972, Kornberg went to the Medical Research Council in Cambridge for postdoctoral work in X-ray crystallography. There he became interested in the X-ray patterns Aaron Klug obtained for chromatin. Using this and other experimental data, Kornberg eventually worked out of the importance of the histones to chromatin structure. Kornberg published his results in 1974. Kornberg stayed on the staff at the MRC until 1975 when he was offered an assistant professorship at Harvard University. In 1978, he moved to the Stanford University, where he is now professor of structural biology. Over the past 35 years, Kornberg has published over 150 peer-reviewed research papers on phospholipid and chromatin structure, gene regulation, and transcription control. His current research interest is on the overall structure of chromatin and chromosomes and how such structuring may be associated with gene repression. In 2006, Kornberg was awarded the Nobel Prize in Chemistry for his studies of the molecular basis of eukaryotic transcription. Kornberg used to play the violin as a child and likes to read. However, he admits that his main interest outside of the science revolves around his family. Kornberg and his research group have made several fundamental discoveries concerning the mechanisms and regulation of eukaryotic transcription. While a graduate student working with Hardin McConnell at Stanford in the late 1960s, he discovered the flip-flop and lateral diffusion of phospholipids in bilayer membranes. While a postdoctoral fellowship working with Aaron Klug and Francis Crick at the MRC in 1970s, Kornberg discovered the nucleosome as the basic protein complex packaging chromosomal DNA in the nucleus of the eukaryotic cells. Chromosomal DNA is often termed chromatin when it is bound to the proteins in this manner, reflecting Walter Fleming's discovery that certain structures within the cell nucleus would absorb dyes and become visible under a microscope. Within the nucleosome, Kornberg found that roughly 200 pairs of base pairs of DNA were wrapped around an octamer of histone proteins. With Yali Lorch, Kornberg showed that a nucleosome on a promoter prevents the initiation of transcription, leading to the recognition of a functional role for the nucleosome, which serves as a general gene repressor. Using this system, Kornberg made the major discovery that transmission of gene regulatory signals to the RNA polymerase machinery is accomplished by an additional protein complex that they dubbed mediator. As noted by the Nobel Prize Committee, the great complexity of eukaryotic organisms is actually enabled by the fine interplay between tissue specific substances, enhancers in the DNA, and mediator. The discovery of the mediator is therefore a true milestone in the understanding of the transcription process. At the same time as Kornberg was pursuing these biochemical studies of the transcription process, he devoted 2 decades to the development of methods to visualize the atomic structure of RNA polymerase and its associated protein components. Initially, Kornberg took advantage of expertise with lipid membranes gained from his graduate studies to devise a technique for the formation of two-dimensional protein crystals on lipid bilayers. These 2D crystals could then be analyzed using electron microscopy to derive low-resolution images of the protein structure. Eventually, Kornberg was able to use X-ray crystallography to solve the three-dimensional structure of the RNA polymerase at atomic resolution. He has recently extended these studies to obtain structural images of RNA polymerase associated with accessory proteins. Though these studies, Kornberg has created an actual picture of how transcription works at a molecular level. According to the Nobel Prize Committee, the truly revolutionary aspect of the picture Kornberg has created is that it captures the process of transcription in full flow. What we see is an RNA strand being constructed, and hence the exact positions of the DNA polymerase and RNA during this process. Kornberg was awarded the Nobel Prize in Chemistry in 2006 for his studies of the process by which genetic information from DNA is copied to RNA, the molecular basis of eukaryotic transcription. Other than the Nobel Prize, Kornberg has received the following awards: 1981 Eli Lilly Award in Biological Chemistry, 1982 Passano Award from the Passano Foundation, 1990 Seba Drew Award, 1997 Harvey Prize from the Technican Israel Institute of Technology, 2000 Gardner Foundation International Award, 2001 Hope Seller Award, Society for Biochemistry and Molecular Biology, Germany, 2001 Welch Award in Chemistry, 2002 ASBMB Merck Award, 2002 Passero Award in Cancer Research, 2002 Grand Prix Charles Leopold Mayer, 2003 elected to EMBO membership, 2003 Marcy Prize from the Keck School of Medicine, University of the Southern California, 2005 General Motors Cancer Research Foundation Alfred P. Sloan Junior Prize, 2006 Dixon Prize from University of Pittsburgh, 2006 to Nobel Prize in Chemistry, 2006 Louisa Gross Howard Prize from Columbia University, 2008 American Philosophical Society membership, 2009 elected a foreign member of the Royal Society. Thank you.